Welcome everyone to this short presentation, which I hope some of you will find very interesting and entertaining. The title of the presentation is called "The Age of Steven Avery's Blood Found in Teresa's Toyota RAV4." And my name is Dr. Silkman, and I'm from the Foul Play team. Welcome. The reason why this area of research is so interesting is that a few years ago, a research group headed by Jonathan Fox did a series of experiments and they published their results in a journal entitled Forensic Genomics. The title of their research paper was Epigenetic Forensics for Suspect Identification and Age Prediction. And this caught my attention as it involves DNA and also the Steven Avery case. So if you can get yourself your hands on a copy of the journal, please do. It is actually quite hard. A lot of libraries don't carry this particular journal. So what did these researchers set out to do? What was their aim? Well, as you know, there's been a lot of controversy in the Steven Avery trial. And of course, what we're looking here is Teresa Halbach. And she was murdered likely on October 31st, 2005. And Steven Avery and Brendan Dassey were found guilty in Teresa Halbach's murder. Now, there's been a lot of controversy, especially when it came to how Steven Avery's blood got deposited inside Teresa Halbach's Toyota RAV4. And during Steven Avery's defense trial in 2007, there was a suggestion that the blood that was found in Teresa Halbach's car that belonged to Steven Avery was planted. So the authors of this paper asked a very simple question. And that question was the 1996 EDTA blood vial, was that particular vial used to plant Steven Avery's blood in Teresa's Toyota RAV4 in 2005. So there was a suggestion by Steven's defense team at that time, Strang and Buting, that law enforcement had taken the 1996 EDTA blood vial and they used some of the blood to plant inside Teresa's Toyota RAV4. Now, this research team, they used a very novel approach. They used a method known as DNA methylation, often abbreviated to DNAm. They used the technique of DNA methylation to essentially to estimate the so-called epigenetic age of blood samples. And this is actually quite novel. So could DNA methylation be used to determine the age of the 1996 blood vial, right? So what is its epigenetic age? And does it actually match the epigenetic age of the blood samples that were recovered from Teresa's Toyota RAV4 in 2005 by Cherie Cohen, who was the DNA laboratory tech who was in charge of all the forensics. So this is a very novel and very interesting approach by Jonathan Fox and his team. Now, DNA methylation itself is a very complicated procedure. Quite a few labs around the world do DNA methylation studies, and it's a specialist area. It's not something that can be done and learned in 5 minutes. It involves a lot of procedures, And there are many, if you're interested in actually learning about DNA methylation, there are plenty of YouTube videos that go through the procedure, and you could check them out. Certainly the molecular biology behind DNA methylation is something that I'm not going to cover in this little presentation. It's way beyond the scope. But let me assure you that there have been many extensive studies that have shown that DNA methylation, now DNA methylation occurs at specific genomic loci. That means we have genes in our DNA where DNA methylation takes place. Now, for those of you who've done a little bit of biology, DNA contains 4 bases, A, G, C, and T. And to control gene expression, in other words, to turn a gene on or turn a gene off, what happens is that at specific cytosine residues, often abbreviated as C, a chemical process that requires special enzymes, those residues become methylated. It's a simple chemical group that is attached to the cytosine residue. And as we age, the DNA methylation pattern in our DNA alters over time. Now, what's important to note here is that the DNA methylation pattern at the specific genomic loci is highly correlated with the age of the cells that you're studying. Now, this type of methylation analysis has been done for blood and urine. So the importance of this, it now enables a research team to predict or make a prediction of an epigenetic age for a specific sample of DNA. So that means that they could take a sample of your blood, a scientist can take a sample of your blood or urine and run it through their molecular biology procedure for DNA methylation, and they can essentially determine the so-called epigenetic age of that sample. For example, if you're 20 years old and you give a sample of blood, then in a perfect world, the epigenetic age of that sample from a 20-year-old person should be around about 20 years of age. Now, there are many factors that affect this, of course. For example, whether you have some type of disease, or whether you drink alcohol, smoke, and also your genetics. There are many factors that can affect epigenetic age. So if you're a 20-year-old and you give a sample of blood or urine and they do DNA methylation analysis, the epigenetic age could be 18, it could range all the way to 22 or 23. So there is a range limit. But if you have a look at the graphs shown up above, you can see that there's an incredible correlation between the so-called DNA age and your calendar age or epigenetic or your normal calendar age, how old you are at the time. So there's a great correlation. That's important, and it's been shown you get this incredible correlation in both blood and urine samples. Okay, now for the researchers, they had to do what is known as a DNA methylation age predictor validation study. They had to show that the analysis that they were doing was reliable and viable. So what did they do? What the research scientists did was they needed to come up with some test to make sure that what they were actually doing was a reliable DNA methylation age predictor. So they generated a mock crime scene, right? that to try and replicate what had happened in the 2005 murder of Teresa Halbach, essentially what occurred in the RAV4. So the research team purchased a 1995 Toyota RAV4, essentially very similar to Teresa's 1999 Toyota RAV4. And what the scientists wanted to do was to generate a mock crime scene. So they got 49 volunteers, and they knew their age, and their age varied between 22 to 80 years. They obtained consent from all the volunteers. They took a sample of blood, and what they did is they deposited the blood in 8 different surfaces in the 1995 Toyota RAV4. So essentially they were trying to replicate the blood that was found in Teresa's Toyota RAV4. So what the scientists did was they placed droplets of blood from the volunteers on the dashboard, the passenger seat, a CD case, the rear door entrance panel, on the console floor, on the driver's seat, on the left passenger seat, and on the ignition casing. And this is exactly what we see in the original 1999 Toyota RAV4 belonging to Teresa Halbach. All right, now the importance of this is that this had enabled the capture and measurement of 513 epigenetic DNA markers. This is important because it now allowed the researchers to draw up a graph, a DNA age versus calendar age graph, which is essentially what you saw in the previous diagram. So now the scientists had this particular graph where they were able to plot all the data points. And that's very, very important. So now the research team have plotted all the data points on a graph. And this is the conclusion that they came up with their data. So their results had shown an ability to find the age of a sample with a strong linear coefficient and a high overall correlation between the known age of a sample and the predicted age across a range of 8 different surface types in the car. And that was really very important, that the data points had shown a very strong correlation. They also then repeated the assay many, many times, and they were able to determine what is known as a technical error. So their data point showed a variation between plus or minus 1.2 years. And when they tested 195 other samples, they had shown that a variance between plus or minus 5 years. It's exactly what I discussed before. So say they took DNA readings, methylation readings from a person who was 22. When they worked out the DNA methylation age or the epigenetic age, some of the samples showed a variation plus or minus 1.2 years, whereas other samples had shown variation plus or minus 5 years. So that way they know the upper and lower boundaries for particular samples. But this was very, very interesting. The research team found that when they looked at all of the samples, the predicted age was not observed to diverge from the actual age of a donor by more than 10 years. That's very interesting. So in other words, they had upper boundaries and likely lower boundaries of 10 years. And you know, when one thinks about it, that is a big potential spread for some of the samples. But as was shown here, the majority of the samples, it was either plus or minus 1.2 years or plus or minus 5 years. But some of the samples had shown a greater variation, but no more than 10 years. All right, so now they validated their research results from samples that they knew the chronological age of the individuals that they tested, right? And they placed the blood on 8 different surfaces. So their model was structurally sound. That way, they can now start testing important blood samples. So let's have a look at what they did. Now, the researchers had used the 1996 blood vial sample, and that was a purple top tube in which the blood was preserved using EDTA. It's an anticoagulant. Now, we know when Steven Avery was born. He was born on the 9th of July 1962. So all the figures that I'm quoting here are the age estimates as determined from the research paper, right? So these are their values. Now, Steven Avery in 1996 was 34.5 years old. The DNA methylation estimate of the 1996 blood vial was worked out to be 37.9 years. So that was an error of 3.4 years. So this is remarkable, right? Because the blood vial had been stored in a styrofoam box for many, many years, greater than 20 years. Yet, the investigators or the researchers were able to take samples of blood from the blood vial and determine that the age, the epigenetic age of that blood vial, the blood in the blood vial, was 37.9 years. As you can see, it was very close to Steven Avery's age in 1996 of 34.5 years. That's remarkable how accurate that is. Now, Steven Avery, I'm not sure is either part of this experiment or for something else. Steven Avery had blood drawn from him in 2016. So the investigators used this blood that was taken from Steven in 2016, and what they did is they placed this blood in and on the so-called mock 1995 RAV4. Now, the DNA methylation estimate was determined to be 52.4 years. Now, Steven's actual age in 2016 was 54.9 years. That's an error of 2.5 years. This, again, is remarkable. It means just by taking blood samples in 1996 and a blood sample in 2016, the investigators found that their DNA methylation estimates were very, very close and accurate. So they worked out that the so-called empirical error range was between 2.5 and 3.4 years. So it looks like their particular testing system was actually quite robust. All right, now here comes one of the most important samples in the Toyota RAV4, and that is the investigators wanted to test item A9, and item A9 is the so-called passenger seat blood spot. Now, this is a blood spot that was cut out of the passenger seat by Cherie Cohen, and she had extracted DNA from that piece of fabric. The investigators wanted to know, could they work out the DNA methylation or so-called epigenetic age from item A9. So, what the researchers found was that the predicted DNA methylation estimate for item A9, again, which is the passenger seat blood spot in Teresa's RAV4, was 52.8 years. Now, what is interesting here was that Steven Avery was 43.4 years at the time of the 2005 murder. He was 34.5 years at the time of the blood being collected in 1996, the purple top tube. This meant, if you look at the DNA methylation age of item A9, this strongly indicated that the blood that was found at the crime scene, that is item A9, and this was identified as Steven Avery's blood, and we'll talk about that in the next slide, it was more likely from 2005 rather than from the purple top tube in 1996. This is very, very interesting. All right, but the complicating factor that pricked up my ears was that the error of this sample was 9.4 years, right? Much higher than previous, than 1.2 or plus or minus 5. This one was actually quite close to their boundary of 10 years. But it's interesting that this sample happened to have an error of 9.4 years. So why is item A9 so important? Well, as you can see here, Cherie Cohen had extracted a genomic DNA from item A9. And she did a PCR analysis, and as you can see, it's an exact match belonging to Steven Avery, right? And the investigators doing DNA methylation analysis have established that that blood is not from the purple top tube. It's got to be closer to when Steven was his age in 2005. All right, so what can we actually conclude from the DNA methylation analysis? This is very interesting and very important. And these were questions that Kathleen Zellner was asking as well. So, point number 1, the passenger seat blood spot, item A9, that was found in Teresa Halbach's Toyota RAV4 at the time of her murder, which occurred in 2005, did not come from the 1996 EDTA preserved blood vial. The DNA methylation result indicated that that DNA sample from the blood was much, much older. So, point number 2, the blood from item A9 was from someone with a DNA methylation age of 52.8 years. Why is that interesting? Well, Steven, at the time of the murder, was 43.4 years old. So there's a discrepancy here. Is it within the limit? Is it out of the limit? It's very, very controversial. But remember, Cherie Cohen indicated that that blood from item A9 was from Steven Avery. It gave his genomic DNA profile. Okay. Now, the researchers, to my knowledge, did not do a PCR profile on that sample, right? They just did a DNA methylation analysis. So this brings up a couple of very intriguing questions. First of all, this is a big one. How did Steven Avery's fresh, non-EDTA blood get inside the Toyota RAV4? That's a really big question. And I think we can conclude that the blood from the sink hypothesis, which I think something that was being pushed by Kathleen Zellner and also by Steven Avery at the time, it's highly unlikely to have occurred. On the proviso, now remember, Kelly Coles has done a series of experiments looking at both fresh blood and blood that came from a purple top tube. And one of the problems with blood that is fresh, that has no EDTA, is that it very, very quickly coagulates and is very, very difficult to reconstitute with water. So it's very difficult to transport and use. The only way that the sink hypothesis can work is literally after Steven had bled in his bathroom sink, the planter or whoever that person was ran inside Steven Avery's trailer, because remember, that's the day both him and Chuck went to Menards. I think it was November the 3rd. Somebody had to have run into the trailer with a pipette, taken the blood from the sink, and immediately went to the RAV4 and planted it. But by doing so, it's extremely risky, because if you don't take the proper precautions, you're likely going to leave your own DNA inside the Toyota RAV4, which is not what you want to do. So, in fairness, is it possible? It is possible. Is it likely? I highly doubt that the sink hypothesis actually took place. Very, very unlikely. And finally, this is an important question. How does the DNA methylation result for item A9 impact Steven Avery's bid for freedom? Because remember, you now have to take into account the DNA methylation age of the blood from item A9. It's clearly not from the purple top tube, but from someone closer to Steven Avery's age. But you've got to take into account that the DNA methylation result from item A9 is unusual, right? It's not closer to 43.4 years, like plus or minus 1.5 or plus or minus 5. It's actually nearly 9.4 years older, right? So that starts to be intriguing. Is there some type of experimental error here? Is it normal? Or is that indeed Steven Avery's blood on item A9, right? These are all questions that need to be asked, and quite clearly, you need to re-extract the genomic DNA from item A9. As an aside, I think the researchers did a brilliant job, but I think they missed out on an opportunity, unless I missed it in the paper. Inside the Toyota RAV4, of course, there was blood belonging to Teresa Halbach, as shown by Cherie Cohen. It's a pity that the researchers didn't do a DNA methylation analysis of the blood that was from Teresa Halbach, because it would have been a perfect internal control, because we knew at the time of the murder, Teresa Halbach was 25 years old. So that would have been very interesting to compare and contrast the results from the Toyota RAV4. So hopefully one day the team or another team that does DNA methylation analysis will try out that experiment. I think it'll be very, very interesting. All right, guys. Well, look, that's really the end of my presentation. It was only a short one. I want to personally thank Yuriz, who's a Foul Play team staff member. He alerted me to the reference, and that was really, really great. And it was a great read, I must admit. At the end of this presentation, please, if you have any questions, leave them in the comment section. I'm just going to post this video up. I'm not going to do a premiere or anything like that. This little video presentation is really just for interest, for those who are interested in the forensics in the Steven Avery case. And guys, if you have enjoyed this little presentation, please subscribe to our YouTube channel and give us a thumbs up. Thank you very much, guys. I'll catch you in the next one.